Hello, science enthusiasts, and welcome to our channel. We often celebrate the titans of science, names like Mendel, Darwin, and Morgan, whose portraits hang in the hallowed halls of history. But what about the brilliant minds who made equally crucial discoveries, only to be written out of the story? Today, we delve into the captivating yet tragically overlooked life of Nettie Stevens, a pioneering American geneticist whose meticulous work in the early 20th century fundamentally reshaped our understanding of life itself. At a time when the very mechanism of sex determination was a complete mystery and the role of chromosomes was fiercely debated, Stevens embarked on a series of groundbreaking experiments that would forever link our identity to tiny thread-like structures within our cells. To truly appreciate the magnitude of her achievement, we have to understand the woman behind the microscope. Nettie Maria Stevens was not born into a world that expected women to become scientists. Born in 1861 in Vermont, she followed a path familiar to many bright women of her era. She became a teacher. For years, she worked diligently, saving every penny not for a dowry or a home, but for a dream, a university education in science. This wasn't a youthful fancy. Stevens was a force of pure determination. She finally enrolled at Stanford University at the age of 35, an age when many of her male colleagues were already established in their careers. She excelled, earning her master's degree before moving to Bryn Mawr College for her PhD, which she completed at the age of 41. Think about that for a moment. She spent decades in patient preparation for a research career that would last only about 10 years. But in that short decade, she would change the world. At Bryn Mawr, she studied under two of the most influential biologists of the day, the future Nobel laureate Thomas Hunt Morgan and the renowned cytologist Edmund Beecher Wilson. These men were giants, and Nettie Stevens, the quiet, older student, was about to make a discovery that would challenge them all. Now let's set the stage. The year is 1905. The scientific world is in a state of exhilarating chaos. Gregor Mendel's revolutionary work on inheritance in pea plants had been rediscovered just 5 years earlier. But his factors of inheritance, what we now call genes, were abstract concepts. No one knew what they were or where they were located inside the cell. The leading hypothesis, championed by Theodore Boveri and Walter Sutton, was the chromosome theory of inheritance, which proposed that these hereditary factors were located on chromosomes. But this was just a theory, and a controversial one at that. Many prominent scientists believed that other parts of the cell, like the cytoplasm, held the secrets to heredity. It was like finding a locked treasure chest, the cell, and arguing over whether the treasure map was written on the gold coins inside, the chromosomes, or was somehow invisibly etched into the wooden box itself. Even more mysterious was the question of sex. How was it determined? The theories were wild and varied. Some scientists argued it was based on nutrition or the temperature of the environment. Others believed it was determined by the age or health of the parents. The idea that something as fundamental as sex could be determined by a specific physical inheritable mechanism was radical. This was the world Nettie Stevens stepped into. Armed with her microscope and an unshakable commitment to observation, she chose a humble subject for her research, the mealworm. Why mealworms? They were easy to raise and, crucially, their cells contained a manageable number of relatively large chromosomes. Stevens wasn't theorizing from an armchair. She was a master of the laboratory bench. For months, she painstakingly prepared slides, staining the cells of male and female mealworms to make their chromosomes visible. Hour after hour, she peered through her brass microscope, not just looking, but seeing. And what she saw was revolutionary. In the cells of female mealworms, she consistently counted 20 large chromosomes. But in the cells of male worms, she found something different. 19 large chromosomes and one conspicuously tiny one. This was the smoking gun. During the formation of sperm cells, the chromosomes would be divided. This meant that half of the male sperm would contain 10 large chromosomes, while the other half would contain 9 large chromosomes plus that one tiny one. The female eggs, in contrast, would all contain 10 large chromosomes. The math was simple and elegant. If a sperm with 10 large chromosomes fertilized an egg, the result was 20 large chromosomes, a female. If a sperm with 9 large and 1 tiny chromosome fertilized an egg, the result was 19 large and 1 tiny, a male. She had found the physical basis of sex. She named the large chromosome X and the tiny one Y. It wasn't environmental, it wasn't nutritional. It was a concrete chromosomal coin flip, and the Y chromosome was the deciding factor. She published her findings in a landmark 1905. Yet history is rarely so straightforward. Her own mentor, E.B. Wilson, published a paper around the same time with similar but less definitive conclusions. And her other mentor, Thomas Hunt Morgan, who was initially a skeptic of the chromosome theory, would later win the Nobel Prize for his work confirming it using fruit flies. In his influential books and lectures, Morgan consistently downplayed Stevens' contribution, often framing her as a mere technician who simply confirmed his and Wilson's more theoretical insights. As a woman without a permanent senior faculty position, her voice was easier to ignore, her credit easier to reassign. Her story is a powerful testament to how a groundbreaking discovery, backed by undeniable evidence, can be overshadowed by the louder voices of the established scientific patriarchy. Her work wasn't just a footnote, it was the cornerstone upon which modern genetics was built. Her story isn't just a discovery, it's a powerful reminder of how easily history can be rewritten, and how crucial it is to seek out the full story behind the science we take for granted. What other forgotten scientists do you think deserve their own spotlight? Subscribe for more hidden histories, and we'll see you in the next one.